Hi, welcome back to Educator.com. This is the first lesson on genetics. Genetics is one of my favorite things to teach in biology because it's all about how you inherited the pieces of DNA that make you who you are. And of course, the environment has a lot to do with the expression of DNA in a person's body or any organism's body. But without that DNA expressing itself, you wouldn't have those raw materials for the environment to act upon. So thanks to the work of Gregor Mendel, a monk from the 1800s, very simple man but intelligent man, we got a great head start with figuring out genetics. So he is considered the father of genetics because his work, his really meticulous, extensive work, really did lay the groundwork. 19th century Austrian monk. He would have spoke German. He lived in what is now Czechoslovakia. The world was a little bit different there in terms of territories back then in the 1800s. But yeah, Austrian monk. Experimented with crossing pea plants in the garden of the monastery. Now, I've asked my students this before, what's the advantage to using peas? Well, if you had tried to do this with animals, there would have been a lot of problems, meaning getting the animals to mate with the ones you want them to, cleaning up after them, the expense of having animals, waiting a long time for the animal to be born, the generation turnover takes a lot longer, the complexity of the genetics of an animal. He got lucky with the pea plants. Pea plants, the species he happened to use, had pretty predictable sort of display patterns for how the genetics is actually shown up, how it shows up in the plant. And you'll see that as we go through some examples with the color of the peas, the shape of the peas, the pod, etc. But this guy, Mr. Mendel, crossed thousands of plants. So this took him years to do. If he would have just mated a few and saw what happened, like, oh yeah, I know what's going on. Well, it was the amount of times he did it to prove that, yes, this can be something that is replicated time and time again, and you get the same kinds of results. That proved his point, his theory about this. So yes, when you look at his data, he crossed thousands of plants. And when we say cross, we mean mated. He took pollen from one, fertilized the flower of the other. And the cool thing about pea plants, in addition to many other advantages that he had, is that every pea plant has male and female parts on it. So that makes it very easy to mate them. And he discovered some consistent patterns based on the results of the crosses. You'll see some interesting ratios of the offspring, what's expected to have happen, when you do these crosses. Thanks to the fact he did it hundreds or thousands of times with these various traits, you get those predictable ratios. And we'll talk more about that, how, flipping a coin 10 times, you're not always going to get 5 heads and 5 tails. But if you flip it 1,000 times, you're going to get closer to what the statistics tell you is going to happen. So there is Mr. Mendel. So with his research, there were 2 laws that have a lot of credence still today, but there are some exceptions that I'll tell you about. His law of segregation. Segregation socially means something very different. If you try to segregate people, you're separating them from each other. Segregation here is talking about gene parts getting segregated. And if you watched the cell division lesson on meiosis, you know that there is a separation of duplicated chromatid parts that has to do with making sperm, egg, pollen. So that's what the segregation is referring to. Mendel knew that each parent has 2 units that it could possibly pass on for each trait, and that these 2 units were split up in their pollen and eggs. There's a 50-50 chance of inheritance. And so you're going to see that depending on the genotype, whether it's dominant, recessive, and that'll make more sense in a little bit, only one of them is going to be passed on to the offspring. Because remember, from the chromosome explanation in cell division, if you have homologous pairs, one from your mom, one from your dad, they each gave you one piece of that puzzle. And Mendel knew that, even though he had never seen chromosomes in his life, he knew that there was something of substance that these plants were passing on in their pollen and egg. So the law of segregation is true. We know it's true. The exception is for when non-disjunction occurs. Non-disjunction would be when you have failure of separation during meiosis, failure of homologous chromosomes to separate or the chromatids to separate. And so you end up with too few or too many chromosomes in sperm or egg or pollen and egg. that doesn't happen that often. So non-disjunction would be the exception to the law of segregation actually happening. The law of independent assortment, a little bit different. Mendel also knew that each trait is inherited independently from every other trait. Example, just because a pea is green doesn't mean it will be round. So the inheritance of color in peas has nothing to do with the inheritance of shape. The size of the plant, whether it's tall or short, has nothing to do with the pea pod color or pea pod look. All those things are inherited separately. The one exception is linked genes. So we now know that when you look at complex inheritance patterns that we know more about today, there are these linked genes where they're found on the same chromosome and tend to be inherited together. One example I can think of is people with red hair are much more likely to get freckles than people with other hair colors. That could be due to linked genes having to do with getting that particular hair color, impacting the expression, the chance of getting freckles. But there are lots of examples of linked genes. Mendel didn't actually get to see that. There was a very simple genetic pattern with pea plants, which I think was a good thing. But you got to cut him some slack. He was doing this work in the 1800s, didn't have a lot of technology, and he really found out some awesome stuff. So some genetics vocabulary. This is the first set of vocabulary I want to share with you, so that as we go through the examples of these different genetic phenomena and these patterns, that we can use these terms and you know what I'm talking about. A gene. Most people know what this is, but it's a segment of DNA that codes for a specific protein. We all know, we get our genes from our parents. What it actually is a segment of DNA. And if you watch the lesson on RNA, you know that RNA is made from DNA and that RNA is translated into protein. And the proteins is what makes the trait. Sometimes these traits are visible on the surface of an organism's body, sometimes they're not, but there is some manifestation of that DNA in the trait. So that's what a gene actually is. An allele is a component of a gene. Organisms typically have two for each gene. They pass on one to their offspring because of that segregation phenomenon. So when we talk about alleles are usually called dominant or recessive. And what that means is, if it's a dominant allele, it pertains to the allele that can overshadow or mask another. So if I inherit two alleles from my parents, and this one is not expressed, but this one is, we would call this one dominant, because it's masking it. It's not allowing this DNA to be expressed. This is the one that's being expressed. Recessive is the one that can be overshadowed. And so these are relative terms. You're going to see there are exceptions to this dominant recessive thing when we talk about more complex patterns. That's coming up a little bit later. Typically, dominant alleles, they use a capital letter. So if we're talking about gene A, dominant alleles would be a capital A. And then for recessive, it would be lowercase. Homozygous and heterozygous. When you inherit two alleles for a gene, the combination is called homozygous or heterozygous. Homo meaning same, zygous coming from that word zygote, which means that first cell of life, the sperm and the egg coming together, or the pollen and the egg coming together. Homo meaning same, so they have the same allele for the gene that is now in that zygote. So homozygous could be this. Or it could be this. Or it could be this. So I'm giving you a few different genes. So for gene A, hey, they're both dominant alleles. Each parent gave one dominant allele. Hey, for gene R, both recessive alleles. And these will be expressed. This is the case where recessive, since no dominant alleles have been inherited, those will get expressed. And here's another example. We would actually call this one homozygous dominant, just to tell us, hey, the ones that are the same, it's the dominant one. This one we would call homozygous recessive, because, hey, the ones that are the same are the recessive ones. Those are the two that were inherited. Heterozygous is having the opposite form of alleles for a gene. So here's heterozygous. This is heterozygous. And that's heterozygous. So the two opposite forms, the dominant and the recessive, inherited together. The mom or dad gave each of them, and That's how it is. You wouldn't use the term heterozygous dominant or heterozygous recessive. That doesn't make sense. Heterozygous tells you you've inherited a dominant from one parent and a recessive from the other. More vocabulary for you. We actually just looked at genotypes on the previous slide. A genotype is the specific combination of alleles for a gene. So examples of genotypes: that. How about this one? Let's go with this one. And I'll give you one that's going to look weird right now, but later on it won't look weird. This is also a genotype pertaining to blood type. But these should look familiar based on what you saw on the previous slide. They're all combinations of alleles that have been inherited. So these are forms of DNA that have been inherited in that new individual. The genotype gets expressed, and the actual physical expression of the genotype is known as phenotype. Sometimes phenotype can be seen when you're talking about something like skin color. Whether or not someone has a widow's peak. If you're unfamiliar with a widow's peak, that's an apostrophe. If your hair comes down to a little point here in the middle of your hairline, that's a widow's peak. As far as I know, that's caused by a dominant allele, if you have it. Another one could be blood type. Now, this is one that you can't tell by looking at someone, oh yeah, you're type A. But there is a physical manifestation, a physical expression of the genes that made this. It's just on the surface of the red blood cell. But all of these are phenotypes, the physical expression of what you inherited up here. Hybrid. There are multiple definitions. The one we're going to use more with this particular lesson is it's an individual who is the result of crossing two sexually reproducing parents. I'm a hybrid of my two parents. That's how you could use it. It also can mean a combination of two closely related species. For instance, a mule is a hybrid, a sterile hybrid of a donkey and a horse. That hybrid thing will come up in biology occasionally, but with this lesson, we're going to talk about a hybrid as a combination of two sexually reproducing individuals. Pure breeding, also sometimes called true breeding, but pure breeding, the individual is homozygous for a gene, only one kind of allele to pass on for a trait. You're going to see coming up when we talk about peas, a pure breeding pea, if it's yellow, it's only going to pass on the alleles that cause its offspring to be yellow. Same with the green pea. If green pea is true breeding, it only has that certain kind of allele. Maybe it's wrinkled, so it's true breeding, or sorry, pure breeding for that wrinkled allele type. All right, so vocabulary associated with the generations. When we cross one set of individuals and get a second group, cross the second group, get a third group, there are names for that. So Mendel started a process for categorizing generations of organisms when tracing inheritance, and he would have kept notes on this to keep it straight, so he knew what generation led to which. The P generation, sometimes called P1, but honestly the 1 is a little bit redundant. P just means parental generation. A, if we plant these peas, we're going to get the next group. So the parents are here. We're saying, hey, this parent is all yellow. I know it's in black and white, so it's hard to tell, but these are all yellow and these are all green. So the parental generation, if we take pollen from one and put it on the flower of the other, the offspring are going to be F1 from the P generation. And F1 is first filial. Filial meaning it's, of the family from the previous generation. And when we cross F1, their offspring are F2, and that pattern continues. If we were to cross F2, the second filial, they'd make F3. You cross F3, they make F4. It keeps going on until F whatever. So yeah, you could think of the P generation as grandparents, and then the F1, the parents, and then F2, the grandchildren. But you know what? It's all relative. F2 is just telling you like, hey, it came from two previous generations. And that's why I wrote and so on. You can keep going if you want to see future patterns coming from all the way back from P. You're going to see that this on the next couple slides that this will make more sense what's going on with this expression, that in the F2, the grandchildren, that one P looks a lot like the grandparent up here. But in the F1 generation, that green phenotype was hidden, it was masked. Okay, Punnett squares. There was a scientist by the name of Punnett who came up with this way of using a square to have a visual representation for the probability of allele inheritance in offspring. Let's say in the P generation, we have a heterozygous individual and we're crossing this individual with another heterozygous individual. So you're going to see this a lot through this lesson. This X just means we're mating those two, and these are their genotypes. The phenotype doesn't really matter here. I just want to show you how the gene inheritance occurs with respect to this Punnett square. So you put one individual on one side and you separate the alleles here and here. This is kind of acting out that segregation phenomenon. During meiosis, you get a splitting of those chromosomes. This is saying that, hey, if that's the male individual, half the sperm are going to have inheritance of this, half the sperm are going to have this other allele in it, or pollen. If we're talking about a plant, half the pollen grains have this, half the pollen grains have that. And the other parent is the same. We put that parent's alleles here and here. This is just how it is. One parent's on the top of this 4-square Punnett square, and the other parent's on the left. You don't put them here or here. That's just how it is. And it doesn't matter where you put the male and female. The male or female could be here and vice versa. And then you show how they come together. It's accounting for all the possibilities of inheritance. So these two gametes, pollen and egg can combine. These two combine. And so on. So what this is saying is, hey, there are 4 possibilities. There's a 25% chance of the offspring being homozygous dominant. There's a 50% chance, 2 out of 4, of it being heterozygous, 25% chance of it being homozygous recessive. So the genotypic ratio for the F1, and if these are the P generation, which are here, this is telling you what's going to happen in the F1 that come from them. So the genotypic ratio is 1 to 2 to 1. That's like saying 25%, 50%, 25%. Here's what I'm saying. I'm saying, hey, 1 to 2 to 1. And this ratio will come up again and again. Some people like writing fractions. It's just another way of representing the chances of this happening. Now keep in mind, if you were to have only 4 offspring, you're not always going to get half of them with heterozygous. Because I mentioned this earlier, if you flip a coin 4 times, are you always going to get 2 heads and 2 tails? No. But if you keep flipping that coin, statistics tell you that as time goes on, you're more likely to get the 50-50 ratio. So if we were doing this with peas and we planted 4 peas to see what happens, well, it's possible we get the perfect 1 to 2 to 1, but every time inheritance happens, every time a pollen grain and egg combine, you get this being reenacted over and over again in terms of the probabilities. So if we planted 100 peas or 500 peas, it's much more likely that it's going to be close to that 1 to 2 to 1 ratio. Okay, monohybrid cross is telling us we're focusing on one gene, mono singular, hybrid combination, cross is the mating. So it's tracing the inheritance of one gene. So Mendel started by mating pure breeding peas in the P generation, no pun intended with the P. So homozygous dominant for this color gene, because he's focusing on color this time. He focused on other traits, the size of the plant, etc. But we're focusing on the color of the P. We're gonna call it the Y gene, and that happens a lot. Geneticists will assign a letter based on the dominant phenotype. You don't have to. I mean, if you want, you could use A for this gene, but we're using Y for yellow. So pure breeding means, hey, this plant, all of its P's are going to look yellow. It's only going to pass on dominant alleles. This plant, all of its peas are green, and because there's no dominant allele, if it was heterozygous, big Y, little y, it actually would look yellow because this allele dominates that. So when you mate them, actually I don't want to use green because I don't want to confuse you about the green pea thing. So here's the pea generation. So this is the pea generation of peas. And when you do a Punnett square, oh, surprise, surprise. They're all going to be heterozygous. And what will they look like? They'll look like this parent, because at least one Y allele, the big one, the dominant one, will mask this recessive one. And so the heterozygous individuals look just like this one. So they're all heterozygous, and they're all going to look yellow. 100% chance. Now, we're going to take these F1 individuals, the F1 all yellow, all heterozygous, and mate them. And I realize that they're pea plant siblings. That's not a big deal when it comes to plants. It's a different kind of thing when it comes to humans, of course. But we're going to mate the F1 generation and see what happens. So for the F1 cross, 2 heterozygotes were mated. Both, well, all of them, are going to be heterozygous looking yellow. So it's going to be a little different this time around. "25% chance of homozygous dominant, 50% chance of heterozygous, and 25% chance of homozygous recessive." So, in the F2, the genotypic ratio is what? It is 1 to 2 to 1. 1 homozygous, 2 out of 4 are going to be heterozygous, 1 out of 4 is homozygous recessive. However, the phenotypic ratio it's going to be a little bit different. It's going to be 3 to 1. Why? Because these 3 all look yellow, but this one looks green, like its grandparent pea plant. So yeah, that's the interesting thing, that now when we look at the genotype versus phenotype, there are some differences, because in this particular case with heterozygous, only the dominant allele is being expressed and the recessive one is being ignored. You're going to see in the future in this lesson, there are some cases where with a heterozygous individual, the recessive is also expressed, but that's not complete dominance like we're seeing here. So there are your ratios in the F2 generation, the grandchildren peas from the P generation with this monohybrid color cross. Now with the dihybrid cross, we're going to trace the inheritance of 2 genes at the same time. That's why it's dihybrid. So Mendel did this. He simultaneously traced pea color and shape. So I'm introducing a new designation for you. Now, as a shortcut, instead of writing, more than this genotype, I'm just going to write big Y blank, meaning that blank, whether it's a dominant Y or recessive Y, same thing in terms of what phenotype you get, because that one dominant is going to be expressed, and whether the other one's a dominant or recessive, it's going to look yellow. Like we also saw before, both recessive, homozygous recessive codes for green, the phenotype. Now with shape of P, getting at least one dominant allele for the R gene is going to make the peas round, like we're used to seeing when we eat peas. But if no dominants are inherited and it's homozygous recessive, you're going to have wrinkled peas. Let's see what happens when you mate the pure breeding plants. So he first did this, he knew, based on previous observation, that, oh, this plant, completely pure breeding. Regardless of what I made it with time and time again, it's always making yellow, round peas. And he mated it with this one. So this one, always yellow, round. Here's the phenotype. And this one, oh, look, it's wrinkled. So it's always making that wrinkled green one. And hey, when you mate them together, we don't even have to do a Punnett Square for this. You're always going to get this. Heterozygous for both genes. And what's it going to look like? Check this out. Look back here. It's going to look like this, like that parent, even though the genotypes are different. So here's what you have in the F1. All of the F1 generation looks like that and has these genotypes. What happens when you mate F1 when it's heterozygous for both genes? You need to do a very large Punnett Square for that. That's coming up next. Here's the Dihybrid Cross example in terms of mating those F1 individuals. So like we said, we're mating this times 2. I'm writing that kind of small because I got to have room to write the different pollen and egg allele combinations you're going to get. So if we account for everything, here's what ends up happening in the pollen grains. That's all the possible combinations. And the way you can keep track of that is, and this is just makes sense in my head, since you have to account for every possibility, I just make 2 little dots next to every letter. And once there's 2 dots, you know you're done. Here's what I mean. So we could have that one and that one combining, right? Okay, that's one pollen grain possibility. We could also have this one and that one. So now we know we're done with the dominant Y. We could also have this one combined with this one. Hey, and we also could have this one combined with this one. And those are all the different possibilities. So since the other parent has the same genotype for both the color gene and the shape of the P gene, it's the same. All right. Now, it would be really exhaustive to write out the genotypes for all of these. So instead, let's just keep track of the phenotypes. Okay, so for the first one, first couple, I'll write the genotype. So yeah, this one is going to look like that grandparent because having that pollen grain combined with that egg, or having, this pollen grain combined with that egg, you're going to get that. And what are they going to look like? Well, going to look like that, right? Now for the rest of them, up on the top row, because of this, the fact that you're always in this part of the Punnett Square, you're always going to get a dominant Y and a dominant R. The rest of these are all going to be round and yellow, regardless of what they inherit here. Even though this particular combination is going to be heterozygous, like those 2 parents, it's still going to be yellow and round. This one, its genotype would be like this. Still, they all look identical. On the same token, this whole left column, all yellow and round. So far, a lot of yellow and round for the same reasons. When this particular gamete is inherited with these, hey, all yellow and round. But hold on, we'll get some varieties. This one right here, you definitely have the yellow color, but oh, little r, homozygous recessive for shape. Looks almost like popcorn, but that's your wrinkled P. This one here, yellow and round. Oh, we have another wrinkled yellow. Yep, because there's no dominant R, but there is a dominant for Y. Next up, Yep. Round and yellow. How about this one down here? Oh, it is also a little popcorn looking one. Has a dominant allele for color, but not for shape. And here's where we finally see some green peas. Check it out. No dominant allele for the Y gene, but it is round. So we're going to have a green round pea like I'm used to eating. Here, what do we have? It's round and green. Different genotype from this one, but same phenotype. And here as well. Oh, and then our last one, completely recessive for both genes. Wrinkled and green. Oh, look at that. It's kind of pretty. Writing a genotypic ratio for this would be kind of a waste of time. It would be a lot of numbers and a lot of colons. It's more meaningful to talk about the phenotypic ratio. So in the F2, for this dihybrid cross, the phenotypic ratio equals, well, how many yellow round peas? 9. How many yellow wrinkled peas? 3. How many round green? 3. And how many wrinkled green? 1. 9 to 3 to 3 to 1. Of course, that number is going to equal 16 because that's all the possible combinations. This is a famous ratio that Mendel came up with. He did dihybrid crosses with a lot of different traits, not just pea color and pea shape. He could have done it with flower position and pea plant size. He could have done it for pea pod shape, pea pod color. The point is that when you do this with a dihybrid cross with both particular individuals being heterozygous in the parental generation, or in this case in the F1 generation, you're going to get this interesting phenotypic ratio. We just talked about complete dominance previously, meaning when you get this, it's going to look yellow, right? This one isn't even expressed. That's known as complete dominance. That does not always happen in genetics. Sometimes inheritance of the heterozygous genotype, both alleles are expressed together. That's called incomplete dominance. Blended inheritance is what another term for it is. Blending of alleles or blending of genes. A good example is snapdragon flowers. So here's a pink snapdragon flower on a snapdragon plant. We're going to start with the P generation. The P generation is going to be, and we use R for red, but this is with the color. The P generation, we have pure breeding red with pure breeding white. So looks red. This one looks white. Now normally if you cross these with complete dominance, all the offspring will be heterozygous and red. But in this case, that's not what happens. So the cross with the P to make F1, all of F1 is heterozygous and pink. They're all pink like this one. So this one's heterozygous. It's definitely a blending, just like if you combine red and white paint, you get pink. So both parts of the DNA on each chromosome are expressed, and you get this collective pigment combination that equals pink. What about when we take the F1 pink individuals, take pollen from one, an egg from the other, and combine them? So here's our F1 cross. Okay, like we saw before, once again, genotypic ratio, 1 to 2 to 1. No surprise there. But what's the phenotypic ratio here? Is it 3 to 1? No, it's not, because these individuals are pink. So both the genotypic and phenotypic ratios for F2 are 1 to 2 to 1. 1 red to 2 pink to 1 white. So there's incomplete dominance. This actually happens a lot in human genetics as well, where there's a blending of traits from both parents to have something that's kind of in between in the offspring. Epistasis. This is a phenomenon in genetics where a gene has the ability to completely cancel out the expression of another gene or genes. It kind of trumps the other genes. Like in certain games of cards, you'll have a trump card where that's the one, hey, I can beat every hand, or I can beat every other card with this. Well, with albinism, the gene that causes albinism to occur, it cancels out all the other genes that relate to skin color. So let's assume there are, let's say there's 4 genes for skin color, okay? Let's say skin color genes A, B, C, and D. But the epistatic gene, the one that can cause albinism, you can see an example here, is gene E, E for epistasis. If you're wondering, like, what literally is albinism, it's the inability to make melanin. So this individual, no melanin. That's the pigment that is in your skin, hair, the irises of your eyes. They just cannot make it in their melanocytes, the cells that would normally produce that pigment. And it's because they've inherited with this gene, this genotype. Homozygous recessive can cancel out all of those. It does, regardless of how much dominant alleles you've got with those 4 skin color genes. So I'm going to give you 3 examples of people with inheritance of these 5 genes, and we'll talk about what they look like. So person number one with skin color, they've inherited this. Now remember, these are the names of the genes, but you inherit 2 alleles for every gene. So there's all 8 alleles for the 4 genes, and they've inherited this. So what do they look like? Are they albino? No. Because of the fact that this is homozygous dominant and not homozygous recessive, you're not going to get those turned off. So what is the skin color like for this person? Fairly fair skin. Not as fair as if they were all recessive, but there's a lot more recessive alleles than dominant alleles. Maybe close to my skin color, it's hard to say, but this person is not albino. How about this individual? Darker skin than the first individual and no albinism. They don't have the homozygous recessive case that would cause them all to be canceled out. So you get expression of these 4 genes. Both of these people, not albino. Now I do want to introduce you to one other term that's going to come up again later in this lesson. The fact that this individual has that 1 recessive allele, they are a carrier for albinism. If they were to mate with someone else with the same genotype, they have a 25% chance of passing on albinism because here's that 1 out of 4 chance that those recessive alleles come together. These 3 individuals, if that actually occurred with their offspring, not albino. They might be a carrier as well, but nope, these people, when you have these genotypes, not albino. This one would be albino. So here's the 3rd example. You can probably see where I'm going with this. Yep, so because of this genotype in the epistatic gene, all of these are not expressed. Now, if they had one of these genotypes, this person would have very dark skin. They have the maximum amount of dominant alleles for these 4 skin color genes. But they're not going to be albino. Or sorry, they are, I misspoke. They're not going to have skin pigment because they're going to be albino because of this epistatic gene. There are also other factors that can impact your skin color, of course. The environment, the amount of sun you get will make melanocytes more active. The food you eat, your health level. There's various factors that can impact the color of your skin, but there definitely are genetics behind it, and this is an example of how one gene can impact a bunch of others. Multiple alleles. We've talked about cases where you have a dominant and a recessive, and those are the only two varieties. But it's a little more complicated than that. With certain genes, there aren't just two forms of alleles, there can be more. An example, human blood types. There are really, really rare blood types, like 1 in a million kind of blood types that I'm not going to discuss here. I'm going to talk about the 4 main letter types. For now, we're going to ignore what's known as the Rh factor. That's whether you're, positive or negative. I'm O positive, so I got a dominant allele that affected the surface of my red blood cells. The Rh factor is inherited via a different gene. So I'm going to ignore that for now. We're going to talk about the 4 main blood types, A, B, O, and AB. So there are 3 allele types here. Here they are. That dominant, this other dominant, and that recessive. What's with the I? Well, this is showing you that, hey, these both are dominant alleles. One is the A form, one is the B form. They are equally dominant over that. That's why they both get a capital I. If instead of this, if we just wrote A and B and wrote little a, you might think that's simpler, but the reason why this is more confusing is you might think that, since that's a dominant B and that's a different letter, it doesn't have the ability to overshadow it like this one does. So to keep it straight, the geneticists who first, came up with this designation, they said, hey, we're going to pick I as the letter representing whether it's dominant or recessive, and we have a superscript saying, hey, this is the A version, this is the B version. They both can mask the recessive here. So with blood types, let's look at if you have phenotype A, the A blood type, whether you're Rh positive or Rh negative, if you're A, there are 2 possibilities with inheritance here. You could be, with your genotypes, homozygous dominant for A, or heterozygous for A. Okay, so both of these people have the same exact red blood cells. Their phenotype is identical. It is complete dominance here with that allele. Now B, you can probably guess what's going to happen. Homozygous dominant for B. Also, they could be B. Heterozygous for B. There you go. With AB, there's only one genotype, and it's this. I've heard this phenomenon being referred to as codominance because both alleles are dominant and they're both being expressed at the same time. Neither one overshadows the other. The combination of these is not a blending like you saw with incomplete dominance. They're both expressed on the surface of the red blood cell. And then there's one other genotype, and it's the one that causes O. To inherit, the homozygous recessive case gives you O blood. Let's do some Punnett squares. Now this is the one for me. This is how I got my blood type. And a lot of times when you do a Punnett square for how you came from your parents, it's hard to know exactly what their genotypes are, but I know for a fact what my parents' genotypes are. And it's not because I did an expensive DNA analysis test or something. I'll explain why I know. So my dad is A, so I know he's got that allele going on. My mom is O. So here's my mom. Here's my dad on the male side, female side. I know for a fact my dad is heterozygous. Excuse me. Why do I know that? Because I got O blood. So if my dad has A and my mom has O, I couldn't have inherited O blood without me getting this allele from him. So yeah, normally I'd write homozygous recessive genotype, but instead this time around I'm just going to give you the phenotype. O and O. There was a 50% chance of me getting O. Turns out that my one sibling, my brother, he's A. So him and I are like a perfect statistical example of how, hey, there was a 50% chance of my parents passing on A blood and a 50% chance passing on O. It's a 1-to-1 ratio, or 2-to-2 if you don't reduce it. Kind of interesting. Yeah, if my dad was homozygous dominant for A, both me and my brother would have A blood for certain. The only way that wouldn't be true is if while I was developing in the womb, there was a random mutation at that exact spot on DNA that codes for this, but the likelihood of that is very, very slim. One more Punnett Square for blood type. I want you to think of this, and if you need some extra time, just pause and look at it for a sec. But I want you to tell me, why is this Punnett Square really interesting? When a heterozygous person with A and a heterozygous person with B blood mate. What's interesting about that? Yep, it's a 25% chance of getting each of the major blood types. Look at that. It's a 1-to-1 to 1-to-1 ratio of all the different blood types. Question for you. Can a blood type be used to prove that someone is the parent? No. To prove that someone is the parent of a child, you'd have to do a more extensive DNA fingerprinting kind of analysis, which you can hear about more with the DNA lesson in this course. But can a blood type be used to prove that someone is not the parent? Yes, sometimes it can. For instance, if we look back at my Punnett Square, if I had B blood, then one of my parents is not the actual parent, because that can't happen. But you can't use blood type proofs that someone is the parent, because there are billions of people, at least hundreds of millions, with that blood type the same as yours. Polygenic inheritance, the name says it all. Poly meaning many, genic, many genes. It's when traits, and there's actually a lot of traits that get this, many traits get their phenotype from the inheritance of numerous genes. So we were looking at a simple, one gene mode of inheritance up until this point. This gene causes this trait, that other gene causes this other trait. But oftentimes, especially when we look at humans, it's numerous genes inherited that together give you a certain phenotype. Human skin color, that's an obvious one. We looked at that particular example with epistasis, that you can have lots of varieties with the number of dominant alleles and recessive alleles. Hair color, there's not just 2 hair colors or 3 hair colors. There's a lot of varieties. And yes, environment can impact that, but hair color definitely caused by more than one gene. And height, there's not just tall and short and medium size. There's a whole rainbow of heights and sizes and shapes with humans. So let's look at height in terms of, why can someone like myself be taller than both parents? I'm about an inch taller, inch and a half taller than my dad, and so is my brother. You can also have cases where the adult child from 2 parents ends up being shorter than both parents, besides, malnutrition and health problems. Sometimes your genes, they give you a predetermined height possibility. How does that happen? Well, let's pick R, S, T, and U. So these are the 4 genes. These are the height genes. It's completely arbitrary why I picked those letters. I just didn't want to do A, B, C, D again. There's T for tall right in there, but let's say there are 4 genes that really impact your height. So here I've got 2 parents, and they're medium size. Let's say the father is 5'9" and the mother is 5'5". Okay. So we've got them both being heterozygous for all 4 genes. And if you're wondering, why did I say that the female is a little bit shorter as average than the male? Well, testosterone, a primarily male hormone, tends to impact growth of men a little bit differently than women. The average height of women is shorter than men. If they mate, chances are, it's more likely they're going to have somebody with a very similar phenotype to them, because they're probably going to inherit a lot of heterozygous possibilities with these 4 genes. However, it's possible they could have a child with this. Look at that. If you do a Punnett Square for each one of these in your head, you can see that for the R, for this to be inherited, because these are the parents, here's the P, and here's the F1. There's a 50% chance of this happening. With S, 50% chance of this happening. With T, 25% chance of that happening. With U, 25% chance. So yes, this is less likely. And you can see that only 2 dominant alleles may be shorter than dad and mom. If this is a boy, it might be the same height as his adult mother. If this is a girl, she might be shorter than mom as well. Here's another possibility. They could actually have a son with this, or a daughter with this. I made this look like, there we go. It looked like lowercase at first, but look at that. This daughter or son has all dominant alleles except for 1. 7 of the 8 alleles, dominance. It's possible. Less likely than some other combinations, but hey, 25% chance of that. 50% chance of the S inheritance. This is 25%, and that's 25%. So chances are they're not going to have a lot of kids like this, but this individual would be taller than both parents. So this is an example of how you can get, this wide variety of phenotypes happening because of polygenic inheritance. With one gene causing one trait to occur, you tend to get less phenotypes. With a test cross, speaking of phenotypes, let's say we have sheep. In the case of complete dominance, so let's say that sheep fur color, with black or white sheep, they have a case where this or this both equal black, and this equals white. When you look at a black sheep, you can't tell what their genotype is, because before I actually listed it as that, I said, hey, that codes for black, right? The fact that you could have homozygous dominant or heterozygous, you can't visually tell. So how do you figure it out without doing an expensive genetic test? How do you figure out what that other allele is? Are they homozygous dominant or heterozygous? Well, you do what's called a test cross. You take a white sheep and you mate them together. If this particular black sheep is homozygous dominant and they mate with the white one, all of their offspring will also have that black wool. You could have them mate 8 times, 12 times, 20 times. You're always going to get black wool offspring. However, if, you can probably guess what's coming here, if they're heterozygous, approximately half will be black and half will be white, because it's a 50-50 chance of inheriting the dominant or recessive with that heterozygous individual. So a test cross is a way to kind of show what genotype is going on there that you can't tell by just looking at the individual. Sex-linked traits. We've talked about a lot of things that have very little to do with male or female, other than the fact that you have a male and female parent. Epistasis impacts men and women equally. Hair color, something both men and women inherit in the same way. But sex-linked traits can impact things that happen in females a little bit differently than what happens in males. Here's some examples. So some alleles are inherited on sex chromosomes. When we talk about the 46 chromosomes in humans, most of them are autosomes or autosomal chromosomes. These are non-sex chromosomes. That would be 22 pairs, which is, of course, 44 chromosomes. There are a total of 46 chromosomes in our species. Most of them have nothing to do with being male or female. However, when you look at the X and Y chromosome, it's one pair. And that's 2 total chromosomes, and that equals the 46. Women are XX, men are XY. There are some other varieties where you can inherit too few sex chromosomes or too many, and that's covered in the second genetics lesson. You can check that out. But X and Y are the 2 sex chromosomes, and actually with cats, they have the same thing going on where XX is a female cat and XY is a male cat. And when we talk about sex-linked, we can also say X-linked. And actually some textbooks and sources you look up call them X-linked. I like to say sex-linked, but it's the same thing. X-linked is a good way to remember it though, because it's only on the X when we talk about these sex-linked alleles or genes. The Y chromosome is really puny compared to the X. So if the X looks like this, actually does look like an X when it's duplicated like the other chromosomes, the Y, oh, so here's the X, there's the Y. It's kind of emasculating the fact that it's so tiny. But this is what a male would inherit. A female would inherit 2 X's. So when we actually say, you know, oh, here's the genotype for a sex-linked trait, it would look like this. So with cats, let's say we're talking about the color. This is a calico cat. Calico cats are only female. It's possible to get a male calico cat, but the male has to inherit an extra X chromosome, which, like I said, if you look that up in the second genetics lesson, it's not a normal male. But for normal sexual chromosome inheritance, calico cats, all female. And here's why. If the gene that causes this coat color, let's call it C, these are all the different genotypes. So this is, of course, equaling the female varieties. With the dominant, the large dominant C allele, that causes black. The small one causes this orange. So in this case, this individual, this female, is all black, so it doesn't have that calico look. This female is all orange, but this one is calico, where you see patches of black and patches of orange. And the reason why you see the patches is in the average female cell, only one X chromosome needs to be expressed. One of them can become what's called a bar body, which is an inactivated X chromosome named after Dr. Barr. So in a calico cat, wherever you see black, this chromosome is active. It is actually being expressed, and this one is turned off. Vice versa with the orange parts. This is the recessive allele on the other X chromosome is active. The other one is dormant. If you're wondering what's the deal with the white, why are there white parts? Well, there's this supposed piebald gene that I've read about. Piebald. That can be inherited, and it can turn off, kind of have an epistatic effect on both of these alleles on the X chromosome. So neither one of those X chromosomes is being expressed in the white patches. So that explains the white along with the orange and the black. Now with males, when you look at the male possibilities, since they're XY, there's only 2 possible genotypes. Because remember, the Y doesn't have that particular allele. This is how you would write the male genotypes. So this male cat would be black, this male cat would be orange. That's it. You're not going to have that patching kind of look because they don't have the 2 X chromosomes. There's an example for sex-linked traits. Now genetic disorders. The genetic disorders I'm going to talk about here are ones that are caused by inheritance of one gene. There are other kinds of disorders, illnesses that are caused by many different genes, and sometimes doctors and scientists aren't completely sure about all the effects So more research needs to be done. But these are disorders inherited genetically. We know it's due to one gene. The first kind I'm going to talk about are autosomal recessive. So these are not on sex chromosomes. They're on pairs 1 through 22, one of the chromosomes there, one of the chromosome pairs. And it's caused by having the recessive, homozygous recessive case. And it's not always going to be the A gene. Like for cystic fibrosis, you'll see F for fibrosis or C for cystic. First one I'd like to talk about is cystic fibrosis. This disorder has to do with a dysfunctional membrane protein that's supposed to shuttle particular ions back and forth across cells, and it's not doing its job very well. It's not doing what it's supposed to do. And that causes pooling of fluids because osmosis makes water go to where there's less water by concentration. And since that membrane protein is not shuttling ions correctly, you get pooling of mucus in lots of different organs. The lungs, the pancreas, in the nasal cavities, etc. And sometimes the organs get so filled with mucus and so damaged that they need a transplant. So the life expectancy for people with cystic fibrosis is lower than others. Now, if you had two people getting together, so this is a, let's call it gene F, okay? If you had two people getting together who were carriers for cystic fibrosis, what is the chance of them having a child with that? It's 25%. So the genotypic ratio, like we've seen before, is 1 to 2 to 1, but the phenotypic ratio, 3 to 1, in terms of their body, in terms of their health. You might say, well, those two people are carriers, but carriers do not have cystic fibrosis. They just have the potential to pass it on. So these three, they all are healthy. This one, that 25% chance, has cystic fibrosis. If this person, if I change it up, was homozygous dominant, they'd have no chance of having a child with the disease because, you would end up having this as the Punnett Square. And then there would be no chance. It turns out that cystic fibrosis is most common in Caucasians. And if you're wondering why is that, there are various disorders that have trends in certain racial groups where they're more common in people of a certain descent versus others. That has a lot to do with what's called the founder effect and human migration patterns. And if you watch the evolution lesson and the human evolution lesson, you'll see more about that. Tay-Sachs disease, let's call it gene T. I could do the same exact Punnett Square as I did before. Tay-Sachs disease has to do with this lipid-based molecule that cannot be broken down effectively in the person's body. And it's because of an enzyme that is not being made correctly due to the wrong, abnormal kind of DNA. And that enzyme not breaking down that molecule, the buildup of it causes severe problems, especially in the brain. And that baby is usually not going to live past the age of 5. So people born with Tay-Sachs don't live long enough to reproduce, but you still have people being born with Tay-Sachs to this day because there are carriers out there. This person is healthy for all intents and purposes. They do not show symptoms because they have this allele on a chromosome and they can make that enzyme. But this person has a chance of making the homozygous recessive individual. So two carriers getting together, it's unlikely it's going to happen, but if they get together and have kids, there's a 25% chance their child will have Tay-Sachs disease. And that's named after the two doctors who discovered it. Sickle cell disease or sickle cell anemia, we could do a Punnett Square for that. We'll use A. Let me actually move this out of the way. Just like before, 25% chance of it happening. And this disease, I mentioned this before, it makes the hemoglobin molecule inside of red blood cells not have the correct shape. And instead of red blood cells being nice and round, they look like this. And they don't carry oxygen as well as this kind. They also get caught in blood vessels and parts of the body and can cause severe complications. Blood transfusion could be a temporary helper, but they're going to continue to make this kind of red blood cell. With PKU, this is known as phenylketonuria. We're going to use gene P for this. That's a mouthful, phenylketonuria, but it's the inability to break down an amino acid known as phenylalanine. The awesome thing about this genetic disorder is that if there's a test done on the baby confirming that indeed they have phenylketonuria, you can avoid symptoms of it as long as you do not feed the child anything with phenylalanine. So do not give them phenylalanine. And by the way, if you watch the RNA lesson, there was a part of the chart, the mRNA codon chart, where you saw PHE, that's phenylalanine. So say no to phenylalanine if your child has phenylketonuria. I know that it's found in chewing gum. There are other foods that have phenylalanine, and as long as they don't consume it, they won't display symptoms of the disorder, which is amazing. So this is one that you can actually control if you have the right kind of environmental exposure. Autosomal dominant, this means that instead of having the homozygous recessive case cause the disease, all it takes is this. So if a parent has it, it's a 50% chance of it getting passed on. So with Huntington disease, let's use H. This individual has Huntington's. This individual does not. The result of the Punnett Square is as such. It's a 1-to-1 genotypic and phenotypic ratio. These people have Huntington's, sad face. These people do not. The reason why I'm assuming that this parent is not homozygous dominant is because that means that both of his parents had to have Huntington's. It would be very rare for two people with Huntington disease to get together and get married and have kids. Very unlikely. So we're assuming he's heterozygous. The mother doesn't have it. The shame about Huntington's disease is that the person typically won't show symptoms until their 40s or 50s, and then it's all downhill from there. You get gradual degeneration, it's a degenerative disease, progressive degenerative disease of the nervous system. And so the nervous system starts breaking down significantly when they reach middle age, and it happens pretty fast, the onset typically. So it's daunting to think about it, that if one of your parents dies from it, there's a 50% chance of you inheriting that harmful allele. And I feel like most people would want to know. They'd want to get the genetic test done to see, hey, do I have the dominant for my parent or do I have the recessive? Polydactyly, let's use a P. This is having more than 5 fingers and 5 toes on a hand or foot. And it is caused by a dominant allele. So if you have a mom or dad with 6 toes, 6 fingers, the other doesn't, once again, it's a 50% chance. There are various communities, the Amish community. I've heard of this mountainous region in Spain, a very isolated community where a lot of the individuals have polydactyly. And when you have a small population with very little immigration and emigration, people coming in and leaving, you tend to have more gene flow between related families. You're keeping those harmful alleles or the alleles you don't want in the population. So with the Amish community, you do have a higher incidence of polydactyly than the whole United States as a whole. So polydactyly, Huntington disease caused by inheriting one or more dominant alleles. Okay, now if we look at genetic disorders that are sex-linked, on the recessive side, there's red-green color blindness. So here's a test to see if you're red-green colorblind. People who can see red and green distinctly could see that there is the number 45 in here because they can tell apart the red and green dots. So if someone's looking at this and they just see a bunch of dots and can't see a number at all, they might be red-green colorblind. And there are actually other color blindness tests that have you tell apart yellow and purple, orange and blue, etc. But red-green colorblindness, one of the more common ones. So the way that it's caused is if you are male, you're going to get that or this, because as I mentioned before with sex-linked traits, there aren't these alleles on the Y chromosome. So this particular individual would not have red-green color blindness, this particular individual would. On the female side, she is not red-green colorblind. She is a carrier, and she actually has it. It's unlikely that a female would be red-green colorblind. It's possible. It's just less common than males because she would have to have a father who's red-green colorblind and a mother who's at least a carrier. But with guys, all it takes is mom passing on that particular chromosome because the Y comes from dad. Here's an example of that. So here's mom, who's a carrier. And here's dad, who has normal vision as well. So mom is not red-green colorblind, but she can potentially pass it on. So here's the Punnett Square. Daughter, normal vision. Son, normal vision. Carrier daughter, son who's red-green colorblind. And it's because mom was a carrier. So there's a 25% chance with this mating or this cross that they're going to have a child with red-green colorblindness. Hemophilia inherited the same way, different gene, different chromosome, but hemophilia is the inability to clot blood. So you have blood proteins that are supposed to come together and kind of form like a net-like structure that catches red blood cells and forms this clot that allows you to not continuously bleed when you've been cut. And platelets would help seal up the blood vessel wall. But hemophilia affects those blood proteins, allowing you to clot. Hemophilia literally means blood-loving. If I were a hemophiliac, I would not love blood, but it just means that the blood keeps coming out. They can carry around clotting factors, or they're going to have to go to an ER if they get a severe cut. So, how does it work? The same kind of way as this other Punnett Square. So here's an example. Let's say that male with hemophilia, female whose father was a hemophiliac, so she's a carrier. Look at this, there's a 50% chance that they're going to have a child with hemophilia. These particular individuals, carrier daughter, healthy male, daughter with hemophilia, son with hemophilia. So those are some Punnett Squares for these sex-linked recessive disorders. Sex-linked dominant, one of the really, really rare kinds of genetic disorders. This means that just having an X chromosome with a dominant allele is going to give you the disease. So you wouldn't have carriers in this case. And actually, I've read that with both of these, you tend to have cases where it's not actually passed on. There's a mutation, a mistake that happens while the child is developing in utero that tends to cause these. But it's possible, it can be passed on by a parent. Rett syndrome, the individual has twitches usually, mental processing difficulties. Microcephaly is a very common trait associated with Rett syndrome. Microcephaly means an underdeveloped brain, literally a small brain. Fragile X syndrome, the X chromosome inherited actually looks like it's about to have a part fall off of it, where It looks like it's kind of deteriorated, or it's going to get detached. And so inheriting this chromosome results in Fragile X syndrome. Males and females can get it. There are cases where the symptoms or the traits can vary a little bit from person to person, but mental difficulty, having a long face, oversized testicles is another characteristic in males. So these are both sex-linked dominant disorders. Thanks for watching Educator.com.